Bacteria make more bacteria, kangaroos make more kangaroos, and dandelions send their fluffy seeds wafting gracefully on the wind to make many more dandelions. But what is it in the seeds that can do that? And how come they always give rise to dandelions and not ever, even once, to Dalmatians or ducks? The answers to those questions lies in deoxyribonucleic acid, or DNA. This nucleic acid molecule carried in the seeds has the complete instructions for making dandelions, which happen to be different from the information for making any other species. Welcome to molecular biology. This is the part of biochemistry that explains how living things reproduce. So yes, this is where biochemistry explains what you've always wanted to know about sex and how you came to be. But in shedding light on how each of us got here, molecular biology also sheds light on all the ways that DNA information can go wrong and what we can do to fix things, perhaps even improve things. Molecular biology brings us in contact with what, at least at the most fundamental level, defines you as a human being and as an individual. Biochemistry traditionally starts with proteins, and proteins are cool, no doubt about it. After all, after using only 20 amino acids, our bodies can build tens of thousands of enzymes and other proteins. And those proteins plus raw materials from food and breathing are all it takes to build all the many other kinds of biomolecules needed by the cell. But where do amino acids and proteins themselves come from? Well, in the nucleic acid that is DNA, we have a single molecule with all the information for making all the proteins in each of the trillions of the body's cells. When you think about what this really means, it boggles the mind. One molecule determines that a child has grandpa's nose, mom's musical talent, and great-grandmother's distinctive cheekbones. On its way to you and me, our DNA has passed through each generation of our ancestors. In each generation, the DNA of two individuals mingled to create unique combinations carried by their children. The fertilized egg that gives rise to each individual contains this DNA, which is copied each time a cell divides, with a copy of the information passed on to every one of the trillions of cells in a human baby. How does a single molecule do this? And how does its structure permit the information in it to be copied easily and accurately when needed? In this lecture, we'll examine the elegant, instantly recognizable double helix structure of DNA and the relation of structure to its function. This will allow us to see how the DNA storage could solve humankind's data archiving problems, and we'll look at how long DNA molecules get packaged into compact chromosomes. Then I will talk about RNA, a close relative of DNA, to highlight how its single-stranded structure gives it greater versatility of function. You'll also learn about how RNA may have played a key role early in the evolution of life. We'll also discuss how viruses get by with very little DNA or RNA. And all this will be in just the first of 12 lectures in molecular biology focused on the wonders of DNA and RNA, how they are expressed in proteins, and the implications of all this for medicine, biotechnology, and more. Our knowledge of how DNA stores information came from its structure. Determination of the structure of DNA in 1953 by Watson and Crick using data from Rosalind Franklin was the most important biochemistry advance of the 20th century. The structure we are most familiar with is a molecule that looks a bit like a rope ladder twisted on itself. Two sides of the ladder serve as backbones of individual DNA strands. Separating the strands, we discover that each strand is a long polymer. The repeating subunits comprising the polymer are called nucleotides. Each nucleotide is made up of three components: a 5-carbon sugar, a nitrogen-containing part called a base, and one or more phosphates. Without phosphate, the resulting molecule is called a nucleoside. Nucleotides that DNA is built from are called deoxyribonucleoside triphosphates, or DNTPs. Deoxyribose, the sugar in DNA nucleotides, has 5 carbons designated 1', 2', 3', 4', and 5'. Why the prime? That's because the base in a nucleotide, which has its own carbons, is also numbered 1, 2, 3, etc. The prime indicates we're talking about the sugar's carbons. The sugar in DNA would be ribose, except that its 'two' carbon lacks the oxygen and has only a hydrogen atom attached. So we call this sugar deoxyribose or 2' deoxyribose. Each nucleotide for building DNA brings energy to the job in the form of 3 phosphates attached to the carbon at the 5' position. Also attached to the deoxyribose sugar is a structure called a base. There are 4 different bases in DNA nucleotides: adenine, guanine, cytosine, and thymine, abbreviated A, G, C, and T. The related molecule called RNA has the same bases, except that uracil, U, substitutes for thymine. 2 of the bases, A and G, are structurally similar and are called purines. The other 3, C, T, and U, are called pyrimidines. Nucleotides get joined together in DNA strands by linking a phosphate to the sugar of an adjacent nucleotide. When the cell builds DNA from nucleotides, the hydroxyl on the sugar's 3' position of one nucleotide is linked to the phosphate closest to the 5' carbon on the next. During joining, the other 2 phosphates from the triphosphate starter material get chopped off, releasing energy to form the bond. Each nucleotide in the finished DNA chain ends up with a single phosphate linking 2 sugars, one sugar from its own nucleotide and the other sugar from the next nucleotide. Linking one phosphate to 2 sugars creates a phosphodiester bond. Esters, let's recall, arise by joining an alcohol to an acid. The acid here is the phosphate group, and it makes bonds to 2 hydroxyls, one on each sugar, whose hydroxyl group functions as the alcohol. Thus, phosphate makes 2 ester bonds, so the bond is a phosphodiester. DNA can contain thousands or even millions of nucleotides strung together in long thread-like structures. Its backbone is made of sugars and phosphates, with the bases roughly perpendicular to the backbone. One end of a strand has phosphates at the 5' carbon of the first nucleotide and is known as the 5' end. The other end of the strand has a free hydroxyl group at the 3' carbon, known as the 3' end. A double helix contains 2 such DNA strands, oriented in opposite or anti-parallel directions, with the 5' end of one strand across from the 3' end of the other. Bases in the 2 strands are oriented such that the A's and T's always face each other, and G's and C's are always across from each other as well. The pairs of bases then form what look like the rungs of a ladder inside of a DNA helix, with sugar phosphate backbones forming the sides. The ladder twists on itself, resembling a spiral staircase. Why do bases pair as they do and never switch partners? It's because the hydrogen bonds between the bases only properly align when G is adjacent to C and when A is adjacent to T. The A's and T's share 2 hydrogen bonds, while G's and C's have 3 hydrogen bonds between them. Since A pairs with T and G pairs with C, it means that if we know the order or sequence of bases in one strand, we can figure out the sequence of bases on the partner strand. It is in the order or sequence of these bases, A, G, T, and C, that information is encoded. Now, what kind of information exactly? Well, one major function of the information in DNA is to provide instructions for making proteins. Proteins are built from amino acids, as you've learned. But how do cells know the order to string the amino acids together to make each protein? It turns out that the information for amino acids arises directly from the sequence of bases in DNA. So the sequence of bases in DNA stores information for making proteins. Information that is securely stored in DNA is copied to RNA whenever the information needs to be accessed and used. RNA's nucleotide composition is the same as DNA, except that U replaces T. So whenever the genetic code is translated to protein, it's in the bases of RNA, which are A, U, C, and G. There are only 4 bases in DNA or RNA, while proteins are being built using 20 different amino acids. Using 1 base or 2 bases to represent 1 amino acid would not work. With 3 bases, though, there are enough possibilities. Cells use 3 bases called a codon to specify 1 amino acid in the genetic code. With 3 bases, there are 4 times 4 times 4 possibilities, so that's 64 different codons. But we only need to make 20 amino acids, not 64, to make proteins. So there are some things to consider. First, in addition to coding for amino acids, the code has 3 punctuation marks called stop codons. These function like periods to signal the end of a protein coding sentence. Second, there is more than 1 codon that specifies most amino acids. This is called redundancy. For example, the 3 base sequences GGG and GGA both specify the same amino acid, glycine. Roughly speaking, the more common an amino acid is in proteins, the more codons there are for it. Almost all the amino acids are specified by at least 2 codons. The codon specifying methionine is also called the start codon because it is almost always the first codon in protein coding sequences. Not every region of DNA codes for proteins, though. All eukaryotic DNAs, for example, including humans, have large sections of non-coding sequences whose functions are numerous and in some cases just beginning to be understood. But every protein coding region in a DNA starts with a start codon, next has a series of codons specifying amino acids, and then ends with one of the 3 stop codons. The rules specifying which codons represent which amino acid are referred to as the genetic code. To see how this works, let's read the amino acid sequence encoded in a stretch of RNA using the codon table here to translate. Remember that the genetic code is only stored in DNA, but is read from RNA where the base U replaces T. We can see the sequence has 5 codons, starting with an AUG methionine start and ending with a UAA stop at the end. In between, we have codons for glycine, cysteine, and glutamic acid. So the linear sequence of bases is read in groups of 3 to get instructions for making proteins. Any region of DNA that contains information needed to make a protein is called a gene, though we will also use a broader definition in later lectures where RNA is the focus. Every protein you've ever learned about, from hemoglobin to the enzymes of metabolic pathways, is encoded in genes. In addition to storing information for making proteins, DNA also contains information that controls when and how much of each protein is made. Topics we'll return to for the translation of genes to proteins in Lecture 31. So we've seen that the structure of the DNA molecule allows it to carry large amounts of information, enough to specify the amino acid sequences of thousands of countless proteins simply by varying the sequence of bases. Let's look at the aspects of DNA structure that allows information to be stored densely and copied readily. Double-stranded DNA molecules have 2 features that make copying it easy. First, as we saw earlier, the base sequences of the 2 strands are complementary. The A's are always connected by 2 hydrogen bonds with T's, while G's are always connected by 3 hydrogen bonds with C's. We call this base pairing. We can see how this works by separating the 2 strands of DNA. When the 2 strands of a DNA molecule are separated, each strand contains information to act as the template for building a new strand. The building of new strands follows the base pairing rules. Adenine always pairs with thymine and vice versa, ditto for guanine and cytosine. Hydrogen bonds between the bases are what hold the 2 strands of the double helix together. The stabilizing force of hydrogen bonds summed together over thousands of base pairs is strong. allowing the double helix to be stable over long stretches. Over short stretches, however, there's only a handful of hydrogen bonds, and those can be broken to separate the strands relatively easily. This is important in the synthesis of both DNA and RNA. United they are strands, divided they fall into being copied. Thanks to the local weakness of hydrogen bonds, DNA strands can be separated relatively easily for replication. This ease of strand separation is important for the enzymes that catalyze copying of DNA. In bacteria, for example, DNA is replicated at the very speedy rate of 1,000 base pairs per second. Now, to get an idea how astonishing this is, consider that a good typist might type 70 words per minute, or about 350 characters per minute. That's just under 6 characters per second. In DNA replication, by contrast, 1,000 fully formed bases are being replicated every second, 160 times faster than our typist. DNA is also a remarkably efficient way to store information. So efficient, in fact, that since 2011, researchers have been working on using DNA to store the enormous flood of data that the world is churning out. The basic idea is simple. Digital data is stored as strings of zeros and ones, like this one. These numbers can be broken up into sets, each of which is represented by a base in DNA. So 00 might be G, 01C, 10A, and 11T. Then once the strings of zeros and ones have been converted into DNA sequence, that sequence can be chemically synthesized and stored. To retrieve the data, the DNA sequence will be read and converted back into a standard electronic form. This is no gimmick. Scientists have shown this is completely possible, and as the costs of the technology drop, it could be one answer to the world's data archiving problems. In 2013, a group of researchers at the European Bioinformatics Institute in England stored all of Shakespeare's sonnets in DNA and retrieved the information without errors. Also, compared to hard drives that can fail in a few years, DNA is extraordinarily stable. Samples of DNA recovered from bones hundreds of thousands of years old are in good enough condition for their sequences to be determined. Once encoded in DNA, data can be stored in a cold, dry place, such as a cave in Patagonia, and it would stay in good condition indefinitely, at least by human standards. It's also easy to have hundreds of copies of the information stored in tiny quantities of DNA, so there would be plenty of backup copies. Despite the enormous quantities of information we are generating, all of the world's data could be stored in a couple of pounds of DNA. DNA is enormous in length, over 7 feet total in each human cell. Very narrow, only 1/25,000th the width of a single hair. This incredibly thin but very long molecule must somehow get stuffed into the nucleus of a cell that you can't even see with the unaided eye. Bacteria have DNA arranged in circular chromosomes. Eukaryotic chromosomes, by contrast, are linear. So eukaryotes, including humans, store their DNA by employing a highly ordered form of spooling. Human DNA is divided into 46 pieces, each of which constitutes a single chromosome. The DNA of each chromosome, which still has millions of base pairs in length, is wound around spools made of proteins. The combination of DNA and proteins is called chromatin, and this combination is what makes up our chromosomes. The proteins making up the spools are rich in lysine and arginine amino acids that are positively charged under cellular conditions. So the proteins called histones are positively charged, and these histone proteins are attracted to the negatively charged phosphate backbones of DNA. The histone core around which the DNA is wrapped contains 4 kinds of histone proteins, known as H2A, H2B, H3, and H4. The resulting complex of protein and DNA under high-powered microscopes looks like a string of beads. Each of the beads is called a nucleosome, And the gaps between the beads, where the DNA is not wrapped around the histones, is called linker DNA or linker region. A fifth type of histone, called H1, is attached just outside the DNA that's tightly wrapped around the histones. The string of bead structures are themselves folded into tightly organized higher order structures. Numerous such levels of tight winding and wrapping create the chromosome structures that we're familiar with. The characteristic stubby chromosomes seen in pictures, however, are the most compact form of chromatin, and that's found only in cells that are about to divide. The rest of the time, the chromatin is not as tightly compacted, which is important for accessing the information in the DNA. What does it mean to access the information in DNA? As we saw, the information for making a protein is encoded in the base sequence of DNA. DNA does not, however, serve as the direct template for building the protein. Instead, the information in the gene is copied into the closely related molecule RNA, which directs the synthesis of proteins. This organization and flow of information is known as the central dogma. A simple statement of it is DNA makes RNA makes protein. DNA making RNA is called transcription. When we state RNA makes protein, it would be more accurate to say RNA codes for protein. The process of converting RNA information into protein is known as translation. Now, RNA, or ribonucleic acid, is the second type of nucleic acid found in cells. Like DNA, it has a backbone of phosphates linking sugars with each sugar attached to a base. In the case of RNA, the sugar is ribose instead of deoxyribose. 3 of the bases in RNA are the same as ones in DNA: guanine, cytosine, and adenine. In RNA, though, thymine is replaced by the base uracil. Like thymine, uracil can base pair with adenine, forming 2 hydrogen bonds. Unlike DNAs, RNAs are made as single strands, which gives them some interesting properties. They can, for example, fold on themselves, with base pairs forming between different sections of the same RNA that are complementary. This is similar to the tertiary folding of proteins. Such folding allows RNA molecules to take on complex 3D shapes, which in turn confer on RNA the ability to carry out functions beyond encoding information. There are 3 major types of RNA in cells. Messenger RNA, or mRNA, is responsible for carrying codon information from making proteins from the DNA to the complexes in the cell known as ribosomes. Ribosomes are the site of synthesis of proteins. Transfer RNA, or tRNA, has 2 roles: carrying amino acids to the ribosomes and providing a decoder of the codons called anticodons. These functions are at opposite ends of the tRNA. One end has a covalent bond to the amino acid, and the other has the decoder. Ribosomal RNA, or rRNA, is a component of the ribosomes that makes proteins. rRNA provides a scaffolding where ribosomal proteins bind and form the overall structures. We'll talk in more detail about all 3 types of RNA in Lecture 30 on transcription. It is in ribosomes that amino acids are joined together with peptide bonds to make proteins. What catalyzes the formation of these bonds? Now, in a big surprise to biochemists, it turned out that the formation of peptide bonds is not carried out by any of the proteins in the ribosome, but by a ribosomal RNA. Now, this is fascinating because it shows that proteins are not the only molecules that can function as catalysts. And how is ribosomal RNA able to be a catalyst? RNA's ability to fold into a complex 3D structure gives it some of the same characteristics as a folded enzyme. RNAs that function like an enzyme are known as ribozymes. It's sort of a mashup of ribonucleic acid and enzyme. So RNA molecules have functions that include catalysis, carrying information, and synthesis of proteins. This diversity of RNA functions helps answer a question that had puzzled scientists for a long time. If enzymes are required to make DNA, And the information for making the enzymes is in DNA. Which came first, the enzymes or the DNA? The so-called RNA world hypothesis suggests that DNA was not the first genetic material. Instead, RNA could have originally performed both functions, replicating itself and catalyzing the formation of peptide bonds to make proteins. In this view, RNA was the molecule from which all of these critical biochemical processes arose. Using selection techniques that allow for what people call evolution in a test tube, catalytic RNAs capable of copying nucleic acid sequences have been created. Now, given the short laboratory time involved in creating these molecules, evolutionary time would clearly be sufficient for creating the variety of RNA catalysts primitive cells might have required. Over time, some of the functions were taken over by molecules that were better suited for particular roles. DNA, for example, is chemically more stable than RNA, and organisms that used DNA as their genetic material likely survived and reproduced more successfully as a consequence. So RNA and DNA are the two types of nucleic acids. DNA is folded for storage into chromosomes, while RNA folding can accomplish enzyme-like functions, very similar to proteins. Now let's discuss viruses, whose sole purpose is replicating their own nucleic acids. Viruses are the ultimate biological opportunists, infecting all known types of cells. Viruses called bacteriophages infect bacteria, and of course, many viruses infect us. Viral nucleic acids code only for a few proteins that each virus needs in its life cycle, starting with the code for their own protein coats. Some viruses also code for their own enzymes for replication, but other viruses hijack any needed enzymes from the cells they invade. This is why an entire virus is physically so much smaller and simpler than a cell, being just a tiny protein particle with a nucleic acid surprise inside. One of the surprises of viral nucleic acids is how many different forms they take. Some viruses contain double-stranded DNAs. Adenoviruses, which cause respiratory infections, herpes simplex, and smallpox are in this category. Others have genomes of single-stranded DNA, such as parvovirus, which infects dogs. Other viruses use RNA instead of DNA to store their genetic information. This can be single-stranded, such as HIV, flu viruses, rhinoviruses causing the common cold, and hepatitis C. Double-stranded RNA viruses also exist, such as rotaviruses, which cause severe dehydration in infants. Viral genomes are sometimes linear, as in the case of adenovirus, or in multiple segments, as in flu virus, or even circular, like bacterial genomes, as in the case of hepatitis C. Sizes of viral nucleic acids also vary enormously. A single-stranded RNA virus like HIV has a relatively small genome size of just over 9,000 bases, whereas the double-stranded DNA virus of smallpox is over 180,000 base pairs in size. Lastly, let's mention that there are actually three common forms of DNA found in cells. bDNA is the most common form of DNA in cells and is the form reported by Watson and Crick in their famous 1953 Nature paper. aDNA is shorter and more compact, with base pairs tilted at an angle to the helix axis. If you're a cell that needs to survive a long time without water, this is the form for you. Now, both of these are right-handed helices that look kind of like this. You could get yourself a job as a graphics proofreader correcting all the images that get this right-handedness wrong in publications. In 1970, a less common form called zDNA was discovered, and it's left-handed. Unlike the right-handed forms, zDNA's cellular function is not yet completely understood. It may be a factor in how some genes are expressed. DNA and RNA probably still have many ways to surprise us. That's why biochemistry is so exciting and fun. DNA and RNA are central to information management of cells and viruses. They really are the blueprints of cells. DNA is largely a repository of the information, while RNA is a jack of all trades, providing an interface between the DNA and the ribosomes that make the proteins. RNA's ability to fold into distinctive structures also is intermediate between the double helix of DNA and the folded structure of globular proteins. Together, DNA, RNA, and proteins remind us of the mantra of biochemistry, that structure is inextricably linked to function. Next time, we'll look more closely at how genetic information is copied and how humans have learned to take copying a step further.